You've probably heard people say things like, men are just more logical, or women are naturally better multitaskers. But what happens when science actually tests those claims? Over the last decade, large-scale brain imaging and meta-analyses have shown that while average size differences exist, the overlap is so great that simple labels like male brain and female brain don't hold up. Many regional differences even shrink or vanish once you correct for total brain volume. And just to be clear, we're talking here about biological sex assigned at birth. Most of these anatomy studies involve people whose gender identity aligns with that assignment, which means the findings don't automatically apply to transgender, non-binary, or intersex individuals. By the end of this video, you'll have a clearer, less binary way to understand cognitive strengths and challenges, and why this question still shows up in everyday conversations. Why this debate still matters in 2025. Walk into almost any workplace, classroom, or family gathering, and you'll still hear people casually mention a male brain or a female brain, as if those categories are fixed. The terms pop up in jokes, dating advice, or quick explanations for why people think differently. Behind these everyday remarks is the assumption that brains fall into neat biological boxes. Yet time and time again, when neuroscience puts those claims to the test, the results throw doubt on the simplicity of those divisions. By 2025, what we know points instead toward a far more complicated and overlapping reality. Most brain imaging datasets have been collected from people whose gender identity aligns with their birth-assigned sex, so we must be cautious applying those results to trans or intersex people. Within those limited samples, one of the most consistent findings has been that, on average, male brains are about 10 to 11% larger than female brains. But this difference mostly tracks overall body size, and it doesn't mean one is better or more advanced. Once size is accounted for, the overlap between individuals is enormous, so large that you can't reliably predict how someone thinks or learns just by looking at whether they are male or female. This single statistic highlights the tension between cultural expectations and scientific evidence. People want clear labels, but the data resists being boxed in. That desire for categories is ancient. For centuries, societies described men as logical and women as emotional, often using those arguments to justify who should lead and who should nurture. Those beliefs shaped education, medicine, and policy. But like many inherited assumptions, they became so familiar that repetition made them feel true, even without supportive evidence. Modern neuroscience has now confronted that legacy by showing that individual brains often mix traits once believed to divide cleanly along sex lines. One person may display structural features considered typical of the opposite sex in older research, and none of that undermines healthy function. This matters because the effects are not only historical, they continue to shape systems we rely on today. In medicine, stereotypes can distort diagnoses. For example, depression has long been considered more common in women, but evidence shows men often present with different symptoms that traditional diagnostic models overlook. Similarly, ADHD has been labeled a male condition, leading to underdiagnosis in women whose symptoms may not fit the expected pattern. Anchoring diagnosis to rigid categories can misdirect treatment, leaving people without the care they need. Drug development has faced similar pitfalls. Clinical trials that assume brain differences are strictly binary may optimize treatment for one group, while failing to detect side effects in others. Dosages based on an imagined typical brain risk ignoring meaningful variation, and create imbalances in how safe or effective therapies are across populations. When the categories themselves are fuzzy, medical research based on those categories becomes less precise, sometimes even harmful. Education offers another clear example. If a teacher assumes boys are naturally stronger in problem solving and girls are naturally more empathetic, the classroom environment itself starts shaping student outcomes. Researchers point out that small behavioral differences can get amplified by adult expectations, creating self-fulfilling patterns. A girl discouraged from pursuing technical fields may internalize limits that began not with her brain, but with how her brain was described. Over time, these cumulative influences can become misread as hardwired truths when they may actually be results of unequal opportunity. Seen in this way, the central question for 2025 isn't whether brains vary between sexes. We know some patterns exist on average. The deeper issue is whether dividing human cognition into two opposing categories adds anything meaningful to science, medicine, or social life. Increasingly, the evidence suggests it does not. Neuroscience reveals tendencies, distributions, and overlaps that are far more complex than the binary framework implies. Averages may exist, but averages also hide enormous variation. The danger of binary labels is that they travel easily, detached from their academic context. A study reporting an average statistical difference between groups can transform into a viral headline declaring that men's and women's brains are different, full stop. On platforms like TikTok or in dinner table conversations, nuance disappears. What began as a cautious scientific observation is reinterpreted as proof of ingrained opposites, fueling stereotypes rather than clarifying them. That dynamic makes careful communication as important as the research itself. So why underline this debate in 2025? Because the costs of oversimplification are real. Misapplied categories warp medical practice, distort education, and reinforce social bias. Meanwhile, the more careful picture drawn from research shows that each brain is a mosaic, complex, variable, and shaped by both biology and experience. Treating brains as if they belonged neatly to male or female boxes actually reduces accuracy, rather than enhancing it. Which leaves us with a natural question. If broad size differences don't tell us much, and binary labels lead us astray, then what features actually matter? To move forward, researchers suggest testing 3 areas of focus that could give us better insight. Whether size versus structural differences make a meaningful impact, how hormones influence brain development and function, and how plasticity allows brains to adapt beyond what biology alone would suggest. And that brings us to the next puzzle. When you place 2 brains side by side, and one is simply bigger, does that difference really mean anything? The size versus structure puzzle. When scientists measure brains, one of the first things they notice is that, on average, men's brains are larger than women's. Large-scale datasets, like the UK Biobank, consistently report about an 11% size difference. That figure sounds dramatic until you account for total body size. Larger bodies require more neural resources to control muscles, organs, and sensory systems, so bigger brains are often proportional to the body they serve rather than a marker of higher capacity. After statistically adjusting for overall brain volume, what researchers call total intracranial volume, many apparent sex-based differences in regional anatomy shrink or disappear. What looked at first like a sweeping biological divide often turns out to be a simple scaling effect. It's also important to understand how misleading averages can be. Even though men's brains are larger on average, the distribution between individuals overlaps so heavily that the trait becomes almost useless for describing any one person. Many women have larger brains than many men, just as many shorter men have bigger brains than taller men. Size may be one of the most frequently repeated facts about brains, but its explanatory power is shallow. Simply pointing to bulk alone tells us very little about memory, reasoning, or creativity. Part of the persistence of the bigger is better myth comes from a false intuition. Larger brains should mean more neurons, and therefore more power. But biology doesn't work like computer processors. A whale or an elephant has a far bigger brain than a human, yet those species don't outperform humans in abstract thought or symbolic language. Within our species, too, sheer size does not map neatly onto greater intellectual performance. The efficiency of connections, not the raw count of neurons, seems to matter most. This is where modern imaging has changed the game. 2 decades ago, brain science had less detail. Researchers could note size, but not easily trace networks. Now, approaches like fMRI and diffusion tensor imaging show how information travels along fiber pathways, how regions synchronize, and how quickly signals move. Brains can be large but inefficient, or more compact yet wired for fast and flexible transfer between regions. These microstructural characteristics, folding patterns, density of neurons, connectivity maps, affect performance far more consistently than bulk volume alone. Cortical thickness has sometimes been highlighted as another anatomical point of difference, with some studies reporting slightly thicker cortex regions in women on average. But here again, the overlap is substantial, and results vary across studies. Researchers are careful not to make categorical claims, because the links between routine anatomical measurements and stable cognitive differences are weak. Outside of injury or pathology, brains of different sizes and cortical profiles generally support the same mental capabilities. In short, there is little strong evidence tying typical structural differences by sex to robust, consistent cognitive outcomes. The broader picture is that anatomy can vary in small group averages, But these distinctions don't translate into fixed psychological categories. A person with a smaller hippocampus can still perform well in memory tasks, and an individual with a larger amygdala does not automatically exhibit higher emotional reactivity. The relationship between region size and behavior is inconsistent across studies, which cautions against declaring that anatomy from one group predicts capacity or temperament for that group as a whole. What researchers emphasize instead is connectivity. Brain networks resemble transport systems in a city, where the placement and efficiency of roads matter more than how much land the city occupies. A densely connected brain can shuttle information with minimal delay, while a larger but loosely wired brain might encounter bottlenecks. These networks don't just differ between people. They adapt across time in response to learning, stress, and experience. Cognitive strengths emerge less from overall mass, and more from the flexible architecture and synchrony that structure supports. This reframing also changes how we treat the public narrative. People often latch onto statements like, "men's brains are bigger," without the necessary qualification that the effect disappears with body size adjustment, and that overlap overshadows averages. In reality, the differences are statistically small, practically limited, and not reliable guides for how someone thinks, remembers, or behaves. The persistence of size-based explanations reflects cultural bias more than scientific utility. Taking apart this myth also undercuts one of the historical arguments used to rank abilities. For centuries, size was conflated with superiority, whether in skull measurements or broader claims about gender capacity. By 2025, science has dismantled that reasoning. What matters is not how much tissue a brain carries, but how effectively that tissue is organized and used. Any attempt to attach intellectual value to sheer volume ends up ignoring the hidden architecture that actually drives mental life. So although brain size often makes the headlines, the weight of evidence shows that structure, wiring, and efficiency matter far more. Size differences exist and can be measured, but they explain little about who we are or what we can learn. For that, neuroscience has shifted its questions away from bulk and toward processes that shape behavior moment to moment. Size may have been the first and simplest way to compare brains, but the real insights come from how networks interact and adapt. And that leads into the next layer of the discussion. If structural traits and connectivity matter more than bulk, what about the chemical signals that steer those networks from day to day? Hormones and the brain's daily shifts. Hormones often become the punchline in casual conversation. If someone gets irritable, distracted, or overly emotional, people quickly chalk it up to hormones. Pop culture has branded testosterone as the aggression hormone and estrogen as the emotional hormone. But the science paints a more intricate picture. Far from being on-off switches that dictate behavior, sex steroid hormones like estrogen, progesterone, and testosterone act as modulators inside the brain. They influence the sensitivity of neural circuits, shape how neurotransmitter systems respond, and fine-tune plasticity at synapses. Their role is less about directly scripting behavior and more about shifting the conditions under which neurons interact. Estrogens, for example, have been shown in animal models and some human studies to promote synaptic plasticity, particularly in brain regions involved in working memory and verbal processing. Specifically, 17-beta-estradiol can enhance the formation of dendritic spines in the hippocampus, supporting networks involved in learning and recall. These aren't dramatic rewrites of how a person thinks, but subtle boosts that create windows of efficiency the brain can take advantage of. Testosterone, meanwhile, interacts with dopamine systems and networks involved in motivation and reward. Rather than forcing aggression or competitiveness, it adjusts the likelihood that certain pathways will activate, depending on context and prior experience. The rhythms of these hormones vary by sex and by life stage. Females experience cyclical fluctuations tied to reproductive biology, including menstrual cycles, pregnancy, and menopause. These cycles introduce changes in hormone concentrations that may influence aspects of cognition and mood over days or weeks. Males, on the other hand, show clearer diurnal variations, with testosterone levels typically peaking in the morning and declining through the day, as well as gradual declines across the lifespan. Both sexes live inside these shifting hormonal landscapes, but the timing, amplitude, and patterns differ. Importantly, neither pattern guarantees fixed behavioral outcomes. Any cognitive shifts are probabilistic and often modest. For instance, studies examining memory performance across the menstrual cycle sometimes report small boosts to verbal memory at points when estrogen is higher. But reviews consistently emphasize that findings are inconsistent. Effects vary from one study to the next, and, when present, are often subtle. In men, higher morning testosterone has been linked to slightly improved spatial or working memory in certain tasks. But here too, the effect sizes are small and not reliably observed across all individuals. These examples illustrate the point. Hormones set conditions that can make some cognitive processes a bit more efficient at particular times, but experience, sleep, nutrition, and stress often outweigh these biological rhythms in practice. This nuance also dismantles older stereotypes. Testosterone can increase the likelihood of competitive or status-seeking behavior, but it does not make a man aggressive in all settings. Social cues and goals matter. Rising testosterone can just as easily fuel persistence in academic work as it can fuel intensity in sport. Similarly, while estrogen interacts with networks involved in emotional regulation and social processing, that does not mean women are destined to be ruled by emotion when estrogen is high. In some studies, estrogen enhances sensitivity to emotional nuance, potentially improving empathy and communication. Attributes once framed as weaknesses are in reality context-dependent strengths or neutral shifts. Clinical science has also shown why these distinctions matter. Early hopes that hormone therapies could neatly preserve cognitive health, especially for women approaching menopause, have not been borne out in large-scale trials. Both the KEEPS and Elite studies, two major investigations into estrogen therapy, found little consistent support for the idea that simply giving estrogen at the right time produces reliable cognitive protection. The mixed results highlight again that hormones are modulators, not magic levers. Cognitive outcomes depend on many interacting factors, and no hormone acts as a direct cure or guaranteed enhancer. An accurate way to picture these effects is not as a switch being flipped, but as adjustments to thresholds. Hormones modulate how excitable certain circuits are, which neurotransmitters are favored, and how easily plastic changes occur. Rising estradiol, for instance, might lower the threshold for encoding new memory traces, while declining levels might reduce that efficiency. A surge in testosterone could sharpen motivation, but whether that motivation is expressed in cooperation or competition will depend on personality, training, and environment. The same biological cue can support multiple behavioral outcomes depending on context. Environment and culture play a decisive role in shaping how these modulatory signals translate into action. Two people with comparable hormonal profiles may respond in different ways because of upbringing, current stress levels, or the cues around them. A rise in testosterone might prompt one person to focus single-mindedly on achieving a personal goal, while another interprets the same biological push as a cue toward interpersonal dominance. Neurobiology provides the backdrop, but environment supplies the script. This more balanced understanding is changing medicine. For decades, pharmaceutical research often ignored or smoothed over hormone-related variability. Trials were scheduled without considering menstrual phases, meaning potential differences in effectiveness or side effects were hidden by inconsistent timing. Now, researchers are beginning to align treatment strategies with these rhythms. The question isn't whether women and men are wired differently. It's how daily or monthly oscillations in hormone concentrations might affect drug metabolism, cognitive performance, or susceptibility to stress at particular times. Mapping treatments onto these rhythms may lead to more tailored, effective care. The same principle applies outside of medicine. Understanding one's hormonal patterns can enable practical adjustments without surrendering autonomy. Some individuals find it useful to track patterns in focus or mood across the month, using that information to match demanding tasks to periods when they feel mentally sharper. Others use awareness of typical dips to practice deliberate coping strategies. Recognizing hormones as moderators of cognitive texture provides a tool for self-awareness, not a strict set of instructions. What matters most is rejecting the outdated idea that biology writes character in bold, unerasable ink. Hormones set the stage for subtle shifts in cognition and affect, but the brain adapts around these shifts with a high degree of flexibility. Cognitive systems recruit alternative pathways, learning refines responses, and conscious strategies regulate behavior far beyond what ebbing and flowing molecules can dictate. Framing hormones as simple causes of personality or mood swings misses both the complexity of neurobiology and the adaptability of human experience. And yet, despite mounting evidence, simplified stories about testosterone and estrogen still influence popular thinking. That persistence isn't random. It reflects a history in which crude generalizations about sex differences were carried into research design itself, affecting what kinds of experiments were conducted and which groups were even included. Those choices left significant blind spots in the scientific record, gaps that still shape medicine and neuroscience today. The hidden gender gap in neuroscience research. For much of modern neuroscience, the picture of the human brain was drawn from only part of the population. Study after study leaned on male subjects, both in lab animals and in people. To most outsiders, this might sound like a technical detail, but it has shaped the evidence base in ways that ripple through how we define disorders, diagnose symptoms, and prescribe treatments. The imbalance was systematic, not incidental, and its consequences remain visible today. In basic research, the gap was especially stark. Early counts of neuroscience studies estimated that male animals were used about 5 times more often than female ones. Researchers argued that by avoiding females, they could control for variability linked to hormonal cycles. The logic was simple. Fluctuating hormones meant more statistical noise, making experiments less clean. What it created instead was a deliberately narrowed dataset that erased half the biology in question. The justification, however tidy, ignored the fact that hormones are part of real life. Cycles of estrogen and progesterone in females are not distractions from brain function, but elements of it. They modulate synaptic plasticity, neurotransmitter systems, and how circuits respond to stress. By filtering out that variation, scientists simplified experiments, but also excluded data that might have revealed different vulnerabilities or protective factors across sexes. For decades, female biology was set aside as a problem to avoid rather than a key part of the neural system worth studying. That decision has had lasting consequences. Medications tested primarily on men often worked differently once prescribed more broadly. Drugs optimized in male-dominated trials sometimes produce stronger side effects or weaker benefits in women. This played out in everything from antidepressants to sleep aids. Dosages that looked effective in trials often turned out less safe or less effective in women. Only after widespread use did some of these gaps become apparent. They weren't mysterious side effects. They were predictable outcomes of limited evidence. Neuroscience, in particular, lagged behind other specialties. While fields like cardiology began to deliberately recruit more balanced samples in the 1990s and 2000s, brain studies clung to older habits. Many labs hesitated to disrupt decades of male-centered data collection, worried about comparability or about the cost of adding analytic layers to handle hormonal changes. Some simply argued the complexity was unmanageable. In practice, this meant conclusions about brain structure and function were comfortable to apply to men, but less accurate for women. The consequences extend beyond drug safety. When diagnostic categories were shaped on male-heavy samples, the typical case often looked male. Conditions such as ADHD or autism were described in ways that aligned with how they appeared in boys and men. Women and girls who showed different patterns, more subtle presentations, different comorbidities, were frequently overlooked or misdiagnosed. Even depression, which occurs at higher rates in women, was initially modeled around male cohorts in trials, blunting recognition of how differently symptoms can manifest. Bias in subjects became baked into the definitions of disorders themselves. Correcting for this decades-long exclusion has only recently begun. In 2016, the U.S. National Institutes of Health introduced its sex as a biological variable policy, requiring researchers to factor sex into experimental design. This move forced labs to reconsider old habits and pushed grant applications to justify imbalances if they persisted. Implementation has expanded the representation of females in both clinical and animal studies, but it has also created new challenges. Handling hormone-driven variability adds statistical complexity, and not every study fully analyzes the data they now collect. Inclusion has improved, but many datasets remain male-biased or treat sex as an afterthought. The culture is shifting, but unevenly. Still, the effort has begun revealing what was missing. Certain patterns now recognized as clinically significant had lurked unseen. For example, some enzymes in the liver metabolize drugs differently depending on hormonal environment, altering how much active compound reaches the brain. Differences once dismissed as anomalies turned out to be common in women, but invisible in male-led samples. Imaging studies are also beginning to detect anatomical subtleties that explain why neurological or psychiatric conditions affect men and women at different rates or in different ways. Each addition makes the overall model more accurate, and each one points to how much nuance was lost by excluding female bodies in the first place. The lag also highlights how hard it is to revisit entrenched assumptions. Entire curriculums, textbooks, and clinical guidelines were built on research that systematically ignored variation between sexes. Rechecking those conclusions takes time and funding, and in the meantime, outdated models still circulate in classrooms and clinics. Students learning from old charts may unknowingly pass forward the very patterns of bias that newer research tries to correct. What looked like consensus in the literature often only reflected male-centered trends. The ethical dimension is unavoidable too. If science positions itself as an objective pursuit, ignoring half the population undermines that claim. Convenience and tradition shaped what kinds of research were considered legitimate. Female biology was not left out by accident. It was defined as deviation from the default male. Recognizing these choices is essential for avoiding similar exclusion under new guises, whether through socioeconomic bias, racial underrepresentation, or assumptions about gender identity. For the audience, this isn't only abstract critique, it affects lived health. Many of the medications in use today were optimized on datasets that underrepresent women. This means dosages may fail to predict sex-specific vulnerabilities or unique side effects. For anyone making medical decisions, it matters that the scientific evidence speaks fully to their body rather than to a population that only partially reflects them. In a practical sense, knowing about the history of research bias can help patients ask better questions and navigate treatment with a clearer sense of possible risks. On the research side, the payoff of inclusion is already visible. By documenting differences in pathways once assumed to be universal, scientists now have a sharper understanding of why disorders vary in prevalence or symptoms between men and women. This growing evidence base not only improves diagnosis, but also guides therapeutic design, making treatments more personalized and effective. The move from avoidance to integration, treating variation as data rather than distraction, is reshaping neuroscience into a field that better reflects actual diversity. The lesson cuts deeper than fairness. When whole populations are excluded, the supposed laws of biology become skewed. Women's side effects, misdiagnoses, and slower recognition of vulnerabilities have all flowed directly from omission. The transition to balanced science isn't only an ethical repair, it's a technical necessity for accuracy. Science cannot describe the brain clearly while half its variations are invisible in the data. And this realization brings us to a second, equally pressing question. Beyond research design, what about the patterns we see in everyday clinical statistics? The striking ways certain mental health conditions seem to cluster differently in men and women. Mental health differences that spark the question. Here's where the puzzle gets sharper. Women show up more often in statistics for anxiety and depression, while men appear more frequently in records of ADHD, autism spectrum diagnoses, and substance use disorders. These aren't isolated quirks in the data, they're consistent trends across decades of research. Still, scientists are careful to point out that these are average prevalence differences, not iron laws. They reflect a combination of biological influences, hormone-linked sensitivities, and social patterns of diagnosis and help seeking. Large research reviews now make a cautious conclusion. Both sex, as a biological category, and gender, as a social system of norms and experiences, contribute, and the relative weight of each varies by disorder in ways that are not easy to measure precisely. For much of the past century, these prevalence gaps were treated as signs of deep biological divides. Higher anxiety and depression in women, for instance, led to speculation that female brains had unique vulnerabilities in mood and emotion circuits. The fact that boys and men were diagnosed more often with ADHD and autism was taken as evidence that male brains were wired in particular ways for attention or social behavior. But that framing skipped over another plausible explanation. Clinicians might be mapping the very same lived experiences into different categories, shaped by expectations of how male or female problems are supposed to appear. Diagnostic bias changed the picture more than most people realized. ADHD offers one of the clearest examples. The disorder was long defined in terms of hyperactivity, children bouncing, blurting, or disrupting class. That image aligned neatly with how ADHD often presents in boys. Girls, meanwhile, were more likely to display inattentive symptoms, daydreaming, quiet spacing out, or subtle disorganization. Because clinicians and teachers were primed to look for the louder symptoms, girls' difficulties often went unrecognized. As a result, men ended up overrepresented in ADHD statistics, not necessarily because the condition is rarer in women, but because the criteria were written around male-centered patterns of behavior. Many women only receive accurate diagnoses as adults, sometimes after years of struggling silently in school or work environments. The same bias often cuts in reverse for anxiety and depression. Women are generally granted more social space to talk openly about sadness, rumination, or stress, so they reach clinicians with symptoms that correspond directly to diagnostic categories. Men, discouraged from presenting those emotions, often express distress through irritability, social withdrawal, or substance use. These presentations can lead providers to record addiction or behavioral disorders instead of mood conditions. In this case, the biology of distress overlaps heavily between sexes, but the pathways into diagnosis fork according to cultural scripts about what men and women are supposed to feel and display. Biology still matters. Neuroimaging shows subtle structural and functional differences in brain regions tied to mood, memory, and reward. For example, women's hippocampal and prefrontal networks sometimes respond differently to prolonged stress, slightly increasing risk of mood disorders in certain contexts. Hormones also modulate these vulnerabilities. Estrogen and progesterone affect serotonin and dopamine pathways in ways that can, during certain phases of a cycle, make anxiety or depressive symptoms more likely to appear. Men, too, live with fluctuating hormones. Testosterone shifts daily and across life stages, shaping motivation, reward-seeking, and in some contexts, risk for addictive behaviors. Peaks can heighten impulsivity, while declines have been linked to negative mood states. But again, the influence is probabilistic, not deterministic. Many men never develop addiction, and many women never experience depression, even though statistics tilt in those directions. Updated studies also highlight that these kinds of hormonal and neural susceptibilities are rarely enough alone. Context, stress load, trauma history, social support often decides whether biological vulnerabilities turn into full disorders. A man with genetically heightened dopamine sensitivity may live without addiction unless combined with high exposure to substances and social reinforcement of risky behavior. A woman with sensitive stress-related circuitry may never experience depression if protected by supportive networks and opportunity for recovery. Biology sets conditions, but environment presses the outcomes. Culture deepens these divides. Men often receive social encouragement toward risk-taking as a tool of expression, while women carry heavier expectations to manage emotions and caregiving roles. These pressures create coping styles, self-medication in men, rumination in women, that strengthen neural patterns over time. By the point of diagnosis, brain networks reflect not only innate sensitivity, but decades of repeated behavior linked to cultural learning. Brain science in 2025 approaches these statistics with that entanglement in mind. Biology cannot fully explain them, but culture cannot either. They interact in loops that spiral across years. The upshot is that prevalence differences don't amount to evidence of two fundamentally distinct brain types. They are the result of multiple overlapping factors: small biological tilts, hormonal fluctuations, gendered expectations, and systemic diagnostic habits that get magnified into neat categories in the data. Understanding how these factors layer together explains why certain conditions appear more common in one group without implying they are hardwired into the brain as male or female destinies. That understanding also matters in clinical life. Misdiagnoses or delays hit hardest at the individual level. A young boy who quietly internalizes feelings may be missed for depression and steered toward discipline instead of care. A woman who struggles with attention, but without hyperactivity, may move through years of frustration without the explanation ADHD could provide. Both are casualties of frameworks built around binary expectations rather than precise mechanisms. Recognizing how prevalence differences emerge helps reduce those blind spots. The direction of current research reflects this shift. Instead of assuming that a diagnosis belongs naturally to one sex, researchers are beginning to isolate the exact factors that matter: which neurotransmitter systems differ in responsiveness, how stress hormones interact with neural circuits, and how cultural experiences reinforce one pathway over another. This approach opens the door to precision medicine, where treatment is guided less by sex category and more by the interplay of hormones, genes, social exposures, and individual neural responses. The key takeaway is clear. Understanding the neural and hormonal bases of sex-linked prevalence patterns is valuable, but applying them responsibly means looking past binary categories. Precision medicine depends on identifying the specific processes, biological and social, that actually drive risks, rather than assuming every man or woman fits an average profile. Seen in this light, the statistics point not to two separate types of brains, but to overlapping systems that display mixtures of traits across individuals, which leads naturally to the next realization. When you examine brains closely, what emerges isn't a split between male and female, but something much more like a patchwork of patterns carried by everyone. The brain is a mosaic, not a binary. Imagine unrolling a quilt stitched from irregular scraps of fabric, some bold and geometric, others muted and delicate, all fitted together into a single design. This image is increasingly how neuroscientists describe human brains, not as two standard templates called male and female, but as mosaics composed of different features, each person carrying a mixture in unique proportions. For generations, the binary model seemed intuitively correct. It offered neat categories and easy stereotypes, simplifying conversations about intelligence, creativity, or behavior. But as brain imaging technology advanced and datasets grew to include tens of thousands of people, the simplicity gave way to greater accuracy. The evidence now indicates that most individuals have a patchwork of characteristics historically labeled male-typical or female-typical, confirming that the brain is not dimorphic, but monomorphic in form. The appeal of binary thinking was its clarity. It suggested that men and women could be separated into two opposing camps of thought and behavior. Men were portrayed as logical, women as empathetic, men as analytical, women as intuitive. These claims were often dressed in scientific language, lending them a sense of authority. Yet research repeatedly eroded the foundation of that narrative. When scientists compared large samples of MRI scans, they found almost no consistent, uniform signatures marking one sex against the other. Instead, brains revealed overlapping designs, collections of features that defied two-category classification and spread broadly across the human population. Specific regions, once thought to be defining markers, provide clear examples. In the hippocampus, amygdala, or corpus callosum, early reports declared sex-linked differences. Larger volumes here for men, increased connectivity there for women. But as data accumulated, overlap dominated the findings. A man could have hippocampal volume within the range, once called female-typical. A woman could display amygdala patterns, formerly stamped male-typical. The more cases studied, the more anomalies appeared, until it was obvious that so-called exceptions were not exceptional at all. Purely male-typical, or purely female-typical brain profiles are rare in large datasets. The typical case is mixed, shaped by many traits combined in non-uniform ways. By 2025, Mosaic findings have become strongly anchored in the literature. Notably, analyses led by Duffner Joel and other researchers combined thousands of scans from international databases and consistently showed that individuals carry a blend of characteristics rather than a uniform type. These multivariate methods revealed the inadequacy of chasing the male brain or the female brain. Instead, they uncovered overlapping distributions, where familiar averages provided only vague shorthand. Binary categories, they concluded, collapse under closer inspection. What we are left with is a model that more accurately describes brains in terms of individual arrangements rather than broad, opposing classes. This mosaic idea reshapes how we think about abilities. Under a binary model, it was natural to claim that men must be better at navigating space, or that women must be more tuned to empathy. But the Mosaic framework explains why those predictions often fail. Within a single brain, features associated with navigation may coexist with features supporting empathy, each present in degrees unrelated to birth sex. Brain function is determined not by box checking, but by how these features combine and interact. Training, practice, and opportunity then shape whether those abilities are strengthened or neglected. A young boy discouraged from exploring emotional expression may underdevelop areas involved in social reasoning, just as a girl pushed away from mathematics may underdevelop parts of her network that support numerical reasoning. Neither limitation was hardwired from the start. Both were possibilities redirected by context. One of the most important contributions of the Mosaic model is how it normalizes variation. Binary categories force contradictions into the margins, treating women who excel at spatial rotation or men who thrive in caregiving as anomalies that need to be explained away. In contrast, the Mosaic approach expects diversity. It predicts that individuals will present scattered patterns of traits, sometimes aligning with group averages, sometimes not. In this light, variety is not an error, it is the expected result. Neuroimaging reinforces this point when viewed across multiple regions, rather than one feature at a time. A single individual may have cortical thickness aligning with female population averages in one lobe, while in another show connectivity strengths aligning with male averages. On large plots of dozens of variables, individuals do not fall into two tidy clusters. They scatter widely, forming gradients without solid borders. The Mosaic framework provides the language that accounts for this scatter more effectively than a binary system could. The consequences extend far beyond research design. Policies in education that continue to tailor teaching methods based on assumed hardwired differences. For example, encouraging boys in technical skills and girls in empathetic ones, are building on inaccurate premises. Corporate training that divides employees into supposedly male strategic types and female collaborative types repeats the same mistake. Even in casual conversation, dismissing behavior as typical of a male brain or female brain misrepresents the evidence. The Mosaic model offers an antidote, emphasizing individuality over stereotype and showing how expectations can misguide opportunity. Another shift highlighted by recent research is the dynamic nature of mosaics. These arrangements are not fixed at birth. Brain patterns change across development as hormones fluctuate in puberty, as experiences accumulate in adulthood, and as age reshapes neural circuits. Plasticity ensures that even if someone's brain at one stage of life aligns more strongly with particular averages, that mix can alter later. Structural adaptations, changing connectivity, or cortical thinning over time all reshape the configuration. A binary model cannot accommodate such change because it assumes permanence. A Mosaic framework, in contrast, expects shifting tiles, rearrangements that span the full length of human life. Seeing brains in this way also changes the tone of interpretation. It removes the burden of supposed deviation. For decades, the binary ideal created pressures to conform. Men avoiding displays of sensitivity to remain consistent with their logic, women avoiding technical ambition to avoid violating their presumed intuition. But science shows that brains simply do not carry these signals. Diversity of traits is the baseline, not a rebellion against it. Recognizing mosaics removes the false idea that someone's brain failed to align fully with their sex. It also reorients identity towards self-understanding. Traits belong to individuals, not categories. For researchers, the benefit is precision. Studying brains through a mosaic lens narrows focus to patterns that actually matter, specific configurations of connectivity, functional responsiveness, or structural resilience, rather than broad and unreliable averages. For society, the benefit is clarity. People no longer need to defend their abilities against a background assumption that sex defines competence. Each person's strengths derive from the unique way their brain is configured and reshaped over time. The image of the quilt remains a useful anchor, but it is no longer a metaphor stretched across paragraphs. A quilt is stitched from many pieces, none dominant alone, each contributing to the whole. Brains are similar, varied elements shaped together, resisting reduction to either or templates. What earlier generations mistook for binary design was the projection of simplicity onto a pattern that was always more complex. Recognizing the mosaic is not about dismantling categories for its own sake, but about replacing weak models with evidence-based ones. Understanding this doesn't only clarify what brains are not, it opens the next frontier. If every person's brain is a shifting arrangement of traits, then the pressing question becomes how those arrangements are shaped over time. Do they reflect innate biology alone, or does lived experience actively rewrite the patterns we carry? If brains are mosaics, the next obvious question is how experience rewires them, which is where neuroplasticity comes in. Neuroplasticity, brains rewrite themselves. Brains constantly adapt to the environments in which they operate. This capacity for change, known as neuroplasticity, refers to the way neural circuits strengthen, reorganize, and sometimes diminish across time in response to new demands. Far from being a fixed machine, the brain is closer to a dynamic system that rewrites itself as people acquire skills, confront challenges, and continue to engage with the world. One striking line of evidence comes from training research. In a landmark set of studies, neuroscientists found that adults who learned new skills, such as juggling, showed measurable increases in white matter pathways and gray matter density related to motor control and visual motion perception. Later work by Scholes, Reed, and Kirby demonstrated that these changes reflected new myelination, the insulating of axons that speeds communication between neurons. What mattered wasn't whether participants were male or female, but the intensity of their practice. Within weeks, repeated activity left traces visible on MRI scans, showing how rapidly plasticity can reshape the brain. For centuries, debates about sex differences leaned heavily on the assumption of hardwiring. Some argued that cognitive abilities were written into the brain at conception, leaving men and women with fixed patterns of aptitude. This framing overlooked the cumulative impact of repeated practice, habit, and environment. Each experience leaves molecular traces, altering synapse strength, pruning unused connections, or encouraging the growth of new dendritic branches. Over time, these traces layer into structure. In other words, biology offers a template, but repeated use is what sculpts the architecture. Education provides one of the clearest demonstrations. For decades, differences in math performance were seen as evidence of innate limits. Girls scored lower on average, and some researchers claimed their spatial reasoning networks were intrinsically weaker. Yet when opportunities to practice mathematics were equalized, the performance gap shrank dramatically. Scans confirmed why. Students who received structured math training developed denser networks in relevant cortical regions, regardless of sex. Apparent aptitude was largely a reflection of how often those circuits were exercised. What had appeared to be innate difference turned out to be differences in access and reinforcement. Athletics tell a parallel story. Young boys often receive greater encouragement for sports that demand coordination, balance, and spatial tracking. Unsurprisingly, brain scans of athletes in fields like tennis or gymnastics reveal strengthened motor and visuospatial networks. But critically, the same changes appear in female athletes trained under similar conditions. Neurons and myelinated pathways expand with practice, not with chromosomes. Gender expectations influenced which circuits were trained, and those choices then solidified into structural differences that looked biological, but were actually cultural investments recorded in tissue. These results highlight why plasticity cannot be ignored when interpreting brain differences. Culture isn't simply an external influence. It is a direct sculptor of neural pathways. Messages such as boys aren't emotional or girls aren't technical are more than stereotypes. They actively restrict practice. Circuits discouraged from use remain underdeveloped, their potential pruned away. By the time adulthood arrives, the gaps are visible on performance tests and brain scans, creating the illusion of fixed sex-linked wiring, when in fact they represent histories of opportunity and denial. Modern research also emphasizes that plasticity is lifelong, though it varies according to age and region. Children display especially high levels of neuroplasticity, capable of rapid adaptation as new skills and languages are introduced. But adults retain significant capacity for change. Studies on language acquisition, memory training, and even recovery from stroke all confirm that new growth and restructuring remain possible well into later decades of life. The rate of adaptation may slow with age, but the principle does not vanish. This means prior experiences and biological differences influence what neural soil is available, but new growth can still occur with sustained effort. Plasticity interacts with, rather than erases, early development. The argument about talent looks different through this lens. Being labeled gifted often reflects an initial advantage in how efficiently circuits adapt to training. But expertise at high levels universally requires thousands of hours of practice. Musicians strengthen auditory and motor areas through endless rehearsal. Mathematicians sharpen problem-solving networks through sustained study. A so-called prodigy is as much a story of exceptional practice opportunities as it is of predisposition. Plasticity explains how those opportunities leave measurable physical traces. Natural differences may influence starting points, but trajectories are powered by use. Neglect can be just as visible as practice. Neuroscientists observe that underused pathways are pruned back, a conservation process aimed at saving energy. If boys are discouraged from developing emotional reasoning, or girls are subtly steered away from spatial tasks, the relevant circuits see less reinforcement and gradually thin. Years later, the absence appears as an entrenched gap in ability, a false confirmation of stereotype. In reality, it was a structural adjustment to repeated lack of demand. Plasticity reveals how inaction, like practice, gets recorded in the folds of the brain. The relevance extends into adult careers and identities. Patterns of employment. Men in STEM, women in communication-heavy fields, look aligned with supposed natural dispositions, but often mirror adolescent histories of training. Brains reflect those paths. The catch is that such paths were constructed by unequal encouragement. People feel pulled toward what they are good at, but what they are good at often originates in what they practiced earliest and most intensively. Seen this way, fields clustered by sex may be less demonstrations of inbuilt aptitude than of experience channeled through cultural norms. Plasticity also reframes clinical practice. Therapists now design cognitive and behavioral interventions aimed at rewiring maladaptive circuits. Repeated exercises can strengthen alternative pathways to regulate anxiety or dissipate destructive thought loops. Neurorehabilitation after injury relies on the same principle. Targeted practice teaches uninjured circuits to compensate. Even pharmacological treatments increasingly pair with practice regimens, so that drugs open windows of receptivity, while repetition consolidates new wiring. Plasticity is not background noise to treatment. It is the mechanism that makes long-term change possible. Pregnancy offers one additional example of structural adaptation. Some research has identified grey matter changes in regions involved with social and emotional processing. The purpose likely reflects preparation for caregiving demands, though findings remain preliminary. Rather than proving universal outcomes, these results highlight the flexibility with which brains respond to major life events, adjusting organization to suit new roles. What emerges consistently is a portrait of the brain as an organ that reflects both biology and biography. Genetics and hormones may set initial conditions, but lived experience, practice, and culture reshape those conditions across an entire lifetime. Recognizing this balance prevents overreach. Plasticity does not erase early patterns, but it ensures they are not destiny. The mixture of inherited template and accumulated activity produces the brain's architecture at any given moment. This understanding carries a sobering implication. If opportunities remain divided, brains themselves will hold those divisions as structural records. If resources are balanced, brains will demonstrate equal capacity to grow in whichever domains are exercised. In each case, plasticity ensures that society's choices about encouragement and investment are literally written into neural pathways. The images of sex-linked destiny lose credibility once cultures are seen as active co-authors of anatomy. By 2025, this conclusion is unavoidable. Plasticity guarantees ongoing potential, but its direction depends on experience. What appears natural may, in fact, be the accumulation of biased practice. The challenge now is to recognize how those accumulated imprints shape what people see as talent in schools, careers, and everyday decision-making. And this is where the discussion turns to real-world consequences. The circuits we reinforce early often influence academic paths and professional futures, creating the impression of innate differences between groups. Understanding how neuroplasticity works makes it easier to see why those differences appear, and why they may shrink when opportunities are equalized. Education and career implications. Ideas about sex-linked ability often show up most clearly inside classrooms, and then continue to echo through career choices. From a young age, children receive cues, sometimes subtle, sometimes explicit, about what they should be good at based on whether they are boys or girls. Mathematics is framed as a male strength, while reading or writing is described as more naturally female. These signals don't come from solid scientific evidence. More often they emerge from cultural expectations reinforced by misread or outdated claims about brain research. Older studies shaped much of this narrative. Small experiments once suggested that differences in brain regions like the hippocampus or parietal lobe might explain stronger math ability in boys and better verbal skills in girls. Those findings were limited. They often ignored key factors such as practice, study time, and the role of encouragement. Despite weak support, the studies were cited widely in education and parenting advice. It was easier to claim boys had natural talent for numbers and girls for words than to disentangle the influence of experience and feedback. The result was an education system where performance gaps became proof of wiring, rather than contexts shaping outcomes. When cultural context is accounted for, the gaps look very different. Large datasets reported in 2025 show that in classrooms where traditional teaching dominates, girls underperform boys in mathematics while boys lag behind in reading. But in classrooms designed to reduce bias and focus on growth, where mistakes are reframed as learning opportunities, the spreads shrink dramatically. Students of all genders score in overlapping ranges, undermining the idea of innate divides. The evidence supports a more cautious view. Group averages can shift depending on environment, and teaching design matters as much as any biological factor. One of the most studied mechanisms behind these shifts is stereotype threat. This describes how reminders of a negative stereotype tax cognitive resources, impairing performance. Laboratory tests and classroom studies alike confirm this effect. When girls take a math exam after hearing cues that girls perform worse, they show heightened stress responses and lower working memory availability, measurable in both physiology and brain scans. That cognitive load suppresses performance, creating apparent deficits unrelated to actual ability. The pattern reverses in reading, where boys reminded that literacy is feminine show reduced engagement and weaker scores. These effects are robust across experiments and highlight how stereotypes operate as an active force in performance, not just background noise. Neuroscience adds further weight. Functional imaging reveals that under stereotype threat, students' brains divert resources away from problem-solving regions towards circuits handling stress regulation. The test becomes not only about solving problems, but battling anxiety triggered by cultural expectation. In contrast, when students are tested without those cues, men and women recruit nearly identical neural networks to solve the same tasks. What looks like brain-based difference is often measurement of stress, not skill. The practical consequences reach beyond the classroom. Early academic experiences carve pathways into later career decisions. A girl who internalizes the message that her struggles with math stem from biology might withdraw from advanced courses, steering away from STEM fields. A boy told that reading isn't a male strength may avoid careers centered around communication or care work. The cycle repeats when lack of representation in one generation discourages a new cohort from pursuing those same paths. Misinterpretations of brain science and rigid belief in natural differences act as gatekeepers, narrowing opportunity fields long before students reach adulthood. Yet the encouraging finding is that these pathways are malleable. Educational programs that emphasize collaborative problem-solving, normalize mistakes, and spotlight diverse role models reduce performance disparities. Teaching practices that frame intelligence as expandable rather than fixed-release students from fatalistic assumptions about what they are capable of. Studies show that when such methods are introduced, the differences between male and female averages shrink substantially. Although small gaps may persist, the sweeping divides suggested by stereotype disappear. This makes clear that group-level differences are not inevitable outcomes of biology, but shifting results of educational design. Careers mirror these educational biases. STEM positions are still disproportionately filled by men, while fields centered on communication and emotional care skew toward women. Many companies explain the imbalance as the result of talent pipelines, claiming men naturally gravitate to analysis, while women choose people-focused roles. But if early, academic separation grows from stereotype threat and biased encouragement, then the adult distributions are more cultural artifact than reflection of destiny. Failing to question those origins risks perpetuating the cycle of exclusion under the cover of biology. The internal consequences matter too. Students and workers internalize limits imposed by assumption. A boy who thinks his writing struggles reflect permanent qualities of a male brain may stop trying to improve. A girl who has been told science isn't natural for her may abandon it, even if her brain circuitry is completely capable of growing with training. Neuroscience shows repeatedly that brains strengthen where they are exercised. Walking away from practice hardens a false perception of limit into reality. The impact on self-belief is where education policy intersects with personal lives. Research documents how strongly mindset influences long-term growth. Students who view skills as trainable rather than inborn show more resilience when facing setbacks and register stronger gains over time. Teachers and parents who adopt this framing can control a powerful variable: the expectation that ability can improve with effort. One specific strategy is to normalize mistakes. Present failures not as proof of lack, but as ordinary steps of learning. Another is to publicly highlight examples of individuals who defied early gaps by persistent practice, cutting across traditional gender categories. These interventions chip away at stereotype threat and give students wider fields to imagine themselves in. Even at the individual level outside formal education, this perspective provides a direct action step. Think back to the messages you received growing up about what you supposedly could or could not do. Identify one area in which you were told you lacked natural ability, whether math, writing, spatial reasoning, or empathy. The science of neuroplasticity suggests trying the opposite. Dedicate deliberate practice to that very skill. If the supposed limit was cultural, the investment will produce visible improvement, sometimes faster than expected. This personal experiment both challenges old stereotypes and makes the neuroscience tangible at a lived scale. By emphasizing practice, context, and expectation, education research reframes the very idea of aptitude. Skills linked to sex are not hardwired features, but responsive, expandable capacities. Classroom design, supportive environments, and individual resolve open space for abilities that seemed out of reach. This fact is liberating because it means that the categories presented as destiny, boys versus girls, logic versus empathy, are historically contingent rather than natural truths. What people can or cannot do is anchored much more in opportunity than in anatomy. These lessons reshape not only schools, but also long-view perspectives about identity and purpose. If differences in learning and talent emerge from the accumulation of experience rather than from binary design, then cultural patterns can be changed and future generations can operate under freer assumptions. This insight stretches into broader life domains, beyond education, beyond work, and into the most fundamental questions people ask about what kind of beings we are. That is where the conversation often shifts, from classrooms and careers to deeper reflections on human consciousness itself. The spiritual side of neuroscience. Spiritual reflection often raises an inevitable question. Do men and women experience inner life differently? Walk into a retreat center or read traditional teachings, and you'll notice references to the masculine mind and the feminine mind. These ideas present consciousness as divided into complementary poles, much like so many other aspects of culture. For some practitioners, the language works as useful symbolism. For others, it risks assigning limits where none may exist. Neuroscience, now placing contemplative practice under the scanner quite literally, suggests a new framing. What we can see in living brains is less about binary pathways of awareness and more about universal networks reshaped by effort and attention. Many traditions have relied on gendered models for centuries. The masculine pole is often described as structured, transcendent, and purposeful. The feminine pole as receptive, embodied, and fluid. But this language is not confirmed in neural data. In fact, studies from 2025 consistently show that dedicated practice engages and strengthens the same core regions across the sexes. The anterior cingulate cortex, important for orienting and sustaining attention. The insula, which integrates awareness of bodily states. And the prefrontal cortex, central for planning and emotional regulation. These findings echo the broader literature on neuroplasticity. Brains do not follow gender-specific scripts, but shift according to how they are trained. That doesn't mean all meditators look identical on a scan. The magnitude of change depends on how long someone has practiced, the style they use, and what kinds of skills they emphasize. A practitioner focused on compassion meditation may show stronger expansions in regions tied to empathy, while someone practicing intensive concentration may display more pronounced stabilization of prefrontal control circuits. Individual personality, motivation, and prior life experience all moderate the effects. Neuroscience shows clear convergence at the level of networks, but also variation across individuals in how far those networks grow. Stress reduction offers one of the most accessible examples. Years of self-reports from meditators claim heightened calm and reduced reactivity after sustained practice. Functional imaging confirms this pattern by showing lowered activation in the amygdala, a hub for processing threat and fear. Importantly, these dampening effects occur in both men and women when practice is consistent. The amygdala does not show a different trajectory by sex. It calms when training targets it, no matter who is doing the training. The shared outcome arises from repetition, not chromosomes. Compassion-focused meditation reveals a similar story. When researchers ask participants to bring to mind care for themselves or others, brain networks linked to social understanding, the medial prefrontal cortex and the temporal parietal junction, activate and expand in response. Men and women show parallel strengthening of these circuits. The idea that empathy can only flourish through a supposed feminine quality collapses in the face of these results. The change isn't sex-specific, it's practice-specific. Anyone who cultivates compassion with enough regularity will leave reshaped footprints in the brain's wiring. This universality is not the same as uniformity. Two practitioners sitting side by side in a hall may walk away with different internal states, not because of gender, but because of the subtleties of their focus and history. One may emphasize quiet attention to breath and report calm. Another may devote more energy to body scanning and report physical sensitivity. Both outcomes are reinforced by practice. What matters is that the broad categories of male mind and female mind add very little to understanding those specifics. Metaphors about masculine versus feminine mind can be useful poetry, but they're not literal brain maps. The most powerful convergence across studies is the remapping of self-regulation networks. Long-term meditators exhibit enhanced connectivity between the prefrontal cortex and subcortical regions like the amygdala. This means conscious control exerts greater influence over emotional reactivity, creating the practical sense of space that practitioners often describe between stimulus and response. It's one of the clearest neurobiological signatures of contemplative training, and it does not differ in kind across sexes. A brain taught to pause before reacting looks like that regardless of whether its owner is male or female. Individual reports reinforce this scientific picture. Sit in on interviews with meditators from different countries, traditions, and identities, and you'll hear the same themes. Sharper attention, steadier mood, a sense of compassion deepening over time. Neuroscience affirms these subjective accounts by documenting the corresponding circuits. And while the magnitude of reported transformation varies along with intensity of practice, the direction points to similar outcomes. The lesson is that consistency, not biology, predicts growth in contemplative capacities. Some nuance remains worth acknowledging. Hormonal cycles and baseline mood differences influenced by genetics can shape how people initially approach practice. For example, stress reactivity thresholds vary across individuals and across stages of life. These differences may color what the first months of meditation feel like. But research shows that with long-term effort, such variations level out into the same pattern of enhanced executive control, improved emotional regulation, and greater empathy. Biology may sway starting points, but practice sets the trajectory. One consequence of this evidence is a shift in what spiritual teachings represent. Historical metaphors about masculine transcendence or feminine immanence continue to inspire, but they should be understood as symbolic rather than literal accounts of the brain. Neuroscience highlights the universality of trainable networks while leaving room for style and variation. The more productive focus becomes, what practice do you sustain, and how do those efforts change your attentional and emotional circuitry over time? In this framing, spirituality is democratically available. Consciousness is not partitioned at the level of sex. Instead, every brain presents the same possibility for transformation, with intensity, method, and persistence shaping outcomes. This realization can be liberating for seekers, concerned that their identity constrains their capacity. The evidence says otherwise. The tools for attentional stability and compassion are present in all brains, and they respond predictably to engagement. From a practical perspective, this opens a different interpretation of what practice means. It is less about becoming a certain type of person, masculine or feminine in consciousness, and more about strengthening common capabilities. When practitioners cultivate focus, the anterior cingulate cortex grows more efficient. When they sustain awareness of bodily sensations, the insula expands its mapping capacities. When they return again and again to compassion, social cognition networks strengthen. These are not symbolic shifts, they are measurable modifications in tissue. Neuroscience therefore restores clarity to the symbolic debates. The binary pathways described by some traditions are not categories written into the brain. They are metaphorical languages that point to qualities available to all. Stated more simply, every contemplative trait can be trained by anyone willing to practice it, because the underlying circuitry is shared human territory. The relevance is direct for anyone listening now. If meditation changes these networks regardless of sex, then contemplating your own path doesn't require worrying about binary models. The critical step is to begin. A single daily routine, 5 minutes of breathing, compassion reflections before sleep. Nudges the same circuits shown in the research. Over time, the nudges accumulate. The brain records each repetition and slowly remaps itself. That process belongs equally to everyone. So here's something concrete you might try. Choose one small daily practice this week and track whether it shifts your mood, focus, or sense of balance. Then share your reflections in the comments for others to learn from. In that exchange, you may find exactly what the data points to: the universality of change, unfolding one practice session at a time. And as we turn from the spiritual to the practical, the question naturally shifts. How can these scientific and contemplative insights be applied in the moments of ordinary life, where memory slips, biases intrude, and relationships test our patience? Practical insights for daily life. So how do you carry all of this into the flow of daily life? The science of brains as mosaics and the reality of plasticity sound abstract until you begin noticing how often you rely on stereotypes to explain away your own challenges or the behavior of others. Applying this knowledge is not about memorizing every detail of hormone research or structural imaging. It's about shifting small, practical choices so that they line up with what the evidence actually supports. One of the simplest steps is to stop using old stereotypes as explanations for your struggles. When you hit resistance, whether in math problems, public speaking, or organizing your time, it's tempting to shrug it off as something your male brain or female brain just wasn't made for. But plasticity research makes clear that ability expands with use. Difficulty usually signals circuits that haven't been strengthened yet, not a hardwired limit. Framing those moments as training grounds rather than proof of incapacity changes how you respond. Instead of giving up early, you understand that the pathway can be built through repetition and deliberate practice. Closely tied to that is how you think about weaknesses. People often assume their blind spots are permanent, painting them as gendered flaws. A man might downplay poor emotional communication by saying, "I'm just not wired for feelings," while a woman might excuse poor sense of direction as female brain stuff. Yet research shows these aren't biological walls. Skills like empathy, spatial reasoning, and memory organization all respond to targeted training. What looks like a weakness is nothing more than a circuit you haven't reinforced as much as others. Reframing weakness as potential for growth takes the sting out of failure and provides a strategy. Practice the neglected skill for a short, regular period and track the change. Bias also creeps into the way evaluations work at school or in the workplace. Teachers and managers often apply different labels to the same behaviors depending on who shows them. A boy's fidgeting can be reframed as creative energy, a girl's fidgeting as lack of focus. A man's assertive tone in meetings might become leadership, while a woman's similar stance turns into being bossy. Knowing that brains are mosaics makes it easier to recognize these patterns as cultural bias rather than objective truth. This gives you room to hear feedback critically, asking whether it reflects your actual performance or whether it's shaped by expectations of what your gender is supposed to look like. Health and diagnosis are another area where practical awareness matters. Because so much past research underrepresented women, certain conditions are still modeled around male-centered presentations. Symptoms for disorders like ADHD, heart disease, or even depression may look different across sexes, yet diagnostic systems often reflect only one template. If you're unsure about medical advice, it helps to remember that diagnostic gaps exist not because biology is simple, but because historical sampling wasn't balanced. Asking second opinions, questioning whether your symptoms fit the classic description, and understanding that bias shapes medical categories can give you more control over your care. Relationships also change when you drop the binary model. Many couples fall into unexamined patterns because they assume one partner brings the logical strengths and the other supplies the emotional balance. But the Mosaic research suggests that both sides contain blends of those capacities. Communication style and problem-solving approach are shaped more by personal history and reinforcement than by sex. Acknowledging this allows couples to approach differences as negotiable habits, not as immovable destinies. Small adjustments, taking turns leading discussions, practicing the habit of expressing emotions directly, can shift long-term patterns that might otherwise feel entrenched. Stress, forgetfulness, and ordinary lapses in daily life can also be reframed. People sometimes joke that being distracted or losing keys is a gendered trait. But these lapses are linked to universal factors like poor sleep, spiking stress hormones, or momentary overload of attention systems. Everyone shares these vulnerabilities. Recognizing that can lighten the frustration by showing you that the slip isn't proof of a faulty male or female brain. It's just part of the universal human condition. A quick laugh at how flimsy the stereotype sounds can even release some of the tension. Humor, when it undercuts rather than reinforces old myths, becomes an ally in navigating those small moments. One way to bring research into your everyday life is to run a personal experiment. Pick a skill you've always assumed doesn't suit you. Perhaps spatial navigation, memory training, or even learning an instrument, and practice it for 15 minutes a day for 4 weeks. Studies show structural brain changes emerge in that time frame. At the end, compare your experience with your baseline. Chances are you'll have improved, sometimes noticeably. That single experiment provides evidence in your own life. The circuits you reinforce expand, regardless of whether people say the skill aligns with your sex. Sharing experiments like this in a community, whether in class discussions, work groups, or even the comments here, allows others to see plasticity in action, not just in data. Here's a concise set of points to keep in mind when putting all of this into daily use. First, don't explain struggles with stereotypes. Difficulty signals training gaps, not fixed blueprints. Second, look at weaknesses as circuits that haven't been built yet. Plasticity means they can be. Third, remember that diagnostic systems have bias. If your experience doesn't fit typical patterns, question whether the template is incomplete. And fourth, choose a small daily practice and sustain it, because measurable rewiring appears in surprisingly short windows of time. Each of these is actionable and grounded in evidence, offering ways to turn neuroscience into tools rather than just abstractions. Stepping back, the broader payoff of applying these principles is freedom. You're no longer tethered to an imaginary template of what your brain should be good at. You're no longer excused from growing because your weaknesses feel biologically inevitable. And you're no longer as vulnerable to biased evaluations that seem to define your limits. Instead, you take responsibility for shaping your own circuits, guided by knowledge that biology tilts odds, but practice writes outcomes. That mindset frees space to pursue your education, career, relationships, and health with clearer options and fewer invisible ceilings. When you think of your brain this way, not as locked into a binary blueprint, but as a shifting mosaic that rewires itself with practice and feedback, the possibilities expand dramatically. And this realization sets the stage for a larger conclusion about what all of this evidence adds up to. The evidence now points in one direction. Brains do not divide neatly into male and female categories. Patterns exist, but averages tell us little about individuals. Large studies show small sex-linked anatomical differences and far greater overlap, with wiring and plasticity, shaped by life experience, carrying the most weight in practice. So here's the challenge. Stop leaning on pseudoscience to excuse stereotypes. A brain is influenced less by sex alone and more by what it practices, what environments it moves through, and what choices repeat over time. It's not defined by sex alone. It reflects a lifetime of biology, context, and practice, and that means it can change. Which skill did you once write off as not for you? And what would you be willing to practice now? Share your reflections in the comments below. Your story may help someone else rethink their own possibilities.